Okay, so we were here and we were talking about how these four phases exist. Now we need to know the details for these four phases. I took 5 minutes after the lecture. I started the Zoom meeting again, couldn't record it, but in that meeting, I asked the students to make sure that when you come today, you're supposed to know the details for all of these, so it's easier for you to process them. And since you've already done it, all of you have, so you don't need to do it again. And then I also asked you to give me 6 reasons why mitosis is important. Now I'm sure Abdullah and Aruba are here, and they'll probably, I'm expecting, because they were here yesterday when it happened, so that I'm waiting for 3 reasons from both of them each. Okay, so let's see, what do we have next. Now we have the details of mitosis. Now, at this point, now everything before this, I wanted to make sure, see, I don't want you guys to remember it or cram it. I just want you guys to understand how the cycles happen. So I gave you a general overview of how the nucleus breakdowns to give us 2 centrioles, and then the whole arrangement takes place, the breakdown takes place, the pulling takes place, and then the cytokinesis finishes after the telophase. Now we need to see these things in details. So we have the interphase. In the interphase, you have the normal cell. How do you know? How will I know that this cell is ready to divide now? 2 things I'm going to see that I talked about the last time. I see replicated chromosomes. I see chromatids that are present in pairs, which mean as chromosomes. The nuclear membrane is still intact, which means the nucleus is not ready to divide yet. And then what's the second thing I see to check that cell division is about to occur? 2 centrioles. Look at them. They have divided and they have started organizing the microtubules around themselves. And we remember that microtubules are essential for cell division. Why? Because those are the ropes that are going to pull the chromosomes apart from the center which we've been calling the centromere, or the place where both of the identical DNAs attach. Let's see, as soon as we end interphase. As soon as we end interphase, what happens is the nuclear membrane starts to disintegrate, which means nucleus is ready to divide. Before this phase, no disintegration, just 2 centrioles. Just as it enters mitosis, the nuclear membrane disintegrates. The chromosomes are so condensed that staining them at this point will help you visualize them individually. Can you see how condensed they are? You can appreciate the centromere. Look at them here. Look how different it is. And I think this picture is either from your book or some updated version of your book. So I want to make sure that you understand every point on this picture and visualize it. So now visualize the nuclear membrane disintegrating. Visualize the highly condensed sister chromatids or the chromosome. Visualize the centrioles moving to different poles. This one's moving away. This one is repelling this one. And they've gathered this microtubule bundle with them to use it. And this bundle at this point is being called the spindle fiber. So at many places in the book, when you read about microtubules, they use words like they're used important because of their ability as spindle fibers, as spindle material, as spindle apparatus. So all of those words simply mean that all of that action of pulling chromosomes apart is responsible to the spindle fibers, which are just microtubules, and being or held by their father microtubule and mother microtubule, which is these two sitting right here, right? Okay, let's see the next step. Once these move to the pole, what happens? Now the nuclear membrane has completely disintegrated. What happens in prophase? Nuclear membrane disintegrates, centrioles move to opposite poles, and chromosomes become highly condensed. In metaphase, these chromosomes are aligned in the center of the cell. Centrioles have moved to polar opposite ends. And what happens is the microtubules extend from the centrioles to attach to the centromeres of the chromosomes. Look at them here. This microtubule attached to this end, this microtubule attached to this end, ready to pull the ropes apart from both ends to separate it from the middle and get this piece of DNA into this side and this piece of DNA into this side. Now, the next time somebody asks you in topic 1, what's the function of centrioles? Will you be able to say that they're responsible for spindle fiber and cell mitosis and cell division eventually? Will you guys be able to say that, or will you guys still be confused about the function of these? Will you guys still say, oh, the only function is cell transport? No, now we know the function is much greater than transport. It's also nuclear and cell division. So I hope this part is clear. Again, metaphase, understand the components of metaphase. Anaphase is just the separation. And how is it the separation? These microtubule spindle fibers have pulled those apart. And how can I tell that they're being pulled apart? Just a simple question, no science here. How can I tell that these spindle fibers were attached to the centromere and not to these chromosome tails? How can I tell by just looking at this diagram? Very easily. How can I tell this very easily? Because imagine this leaflet and the center of it. If I tied my rope here and pulled it from here, how is it going to be pulled towards me? It's going to be pulled like this, right? It's going to move towards me like this. But if I'm attaching my rope here and pulling over here, then how it's going to be pulled? It's going to have its center here and its wings flapping back. Have you ever pulled a kite in your life? If you have, then you will understand when you're tugging at the kite, when you're pulling it towards yourself, the center that you're using to pull it towards yourself moves towards you first. If in this chromosome, if I tie my rope here, what's going to happen? This end is going to move towards me first. The rest of it will follow. So the chromosome is so smart. It does not want to see, because all of these phases are occurring so quickly. Look at this. The cell is bending inwards. The phase of cytokinesis is very near. Now, the cell does not want that. So let's imagine this is cytokinesis very near, and you're pulling on it from this end, and this end remains behind and gets cut off by chance. If this process becomes the cytokinesis becomes faster, then you'll only get this part into the cell and the rest of it will remain behind. So the cell is clever enough to pull at the right place, which is the centromere. If the microtubule rope is tied around and it's pulled towards the MTOC or the centrioles, to pull it in the shape, in such a shape that the center moves in first and both of those parts are equally pulled in towards the centriole. Is this clear? And why am I explaining such an insignificant point? Because in my paper, I still remember there was this one question where they asked you to draw, they gave you the metaphase and they asked you, okay, draw the anaphase. And people went about drawing it as single chromosomes being here and single here, but they were all marked wrong. The right answer was this exact shape in which the centriole is placed like this and the tails are behind it flapping. Why? Because they have nothing attached to them. They're free. Only the centromere is being pulled in, and the tails, these chromosome tails, are behind it, flapping behind. Do you understand what I mean? What I said,
Yes,
is
teacher.
it clear
Yes.
for
Yeah.
everyone? So you will
Yeah,
see that
miss.
question, and I want to make sure that you do not miss it, because that question can be manipulated in so many ways. They can, for instance, not give you a centrioles and not give you an entire cell and be like, okay, tell me where the centrioles are. Are they on position A, that is this, or are they on position B, that is this? Now, if you don't know the answer, you can get confused. You know that the centrioles are where the place from which the rope is going to be attached to the centromere. So the centromere will be pulled in first. So obviously position A, or this position, is where the centromeres are, and not this position where the tails are lying first, right? Just so you guys have it clear. Okay, and then how do you appreciate an anaphase? You appreciate it by appreciating this pulling apart. But when you realize that the moment these centromeres separate and we get two chromatids separately, is when the anaphase starts, because it's occurring so simultaneously, so quickly, it can be difficult to appreciate. But like I told you, these condensed chromosomes are what we stain in when we're staining cells. It's very easy if we make freeze sections of these very quickly. It's very easy to look at these sections, which are given in your book and which I'll also talk about. So what is the telophase? It's the exact phase in which the nuclear envelope now begins to reform We have single chromatids in each nucleus, and decondensation of these will take place as soon as the nuclear envelope completely forms, and pinching inwards takes place. This pinching will be completed in cytokinesis. Okay. In plant cells, so I couldn't find I did not find a very good diagram. I'll try to look for it, but just to tell you over here, in plant cell what ends up happening is, for instance, if this is mitosis in a plant cell, these are the centrioles, these are the nuclei, this is the centrioles, the nuclei. In these, cytokinesis is not pinching inwards because this is covered by a cell wall. It's very difficult to pinch a cell wall inwards. So what ends up happening is that the Golgi come and deposit over here, and these Golgi synthesize cellulose inside them by the help of endoplasmic reticulums, and those carbohydrates are synthesized to form a cell membrane exactly in the center of the cell. And then the membrane of the Golgi is obviously the cell surface membrane, which forms the cell surface membrane of this cell and this cell. And the internal cellulose cell wall that forms between these two cells is thanks to the Golgi apparatus and the functioning of the rough endoplasmic reticulum. So is the cytokinesis of the plant cell also clear? It's very different from this one. This one is just a pinching inwards movement of the cell membrane, but that is not possible here due to this cell wall. Okay. And then so one thing I did wrong here is that I drew centrioles for the plant cell, which I also forgot, but there are no centrioles. Nobody pointed it out. I've been telling this again and again. There are no centrioles in plant cells. No centrioles at all. Plant cell mechanics are much more complicated than an animal cell. There are no centrioles. There is still this spindle fiber forming, but it's thanks to other organelles that are not in your syllabus, but there are no centrioles. So is this clear?
Yeah, miss.
If even for
Miss?
Yep.
Yes.
Miss, I was doing a question, past paper question, and it asked about the cytokinesis of plants, and it had the marking scheme had cell plates in it. So can you tell about cell plates?
Okay. So exactly like I said, exactly like I said, if this is your plant cell, these are the two nuclei separated. When this Golgi comes in and deposits over here to start making the cell wall material, this red one inside, and more Golgi, see, Golgi are separate apparatus, right? More Golgi form. So this apparatus that forms thanks to the Golgi with the synthesis of the cell wall inside, this is known as the cell plate apparatus because it's like a single plate. It's actually the Golgi apparatus and some other organelles also coming in to form this structure inside, which is not exactly pinching. This one is pinching. This one is deposited structure of the Golgi. This is known as the cell plate. Is that clear? It's called a plate because it's depositing it's not depositing linearly. For instance, some Golgi deposits over here, some over here, some over here, and eventually when these join together and these are plates, when these join together, so the cell will be divided into two. So it might not be identically in the center, it can be a little bit skewed as well, which is why we call it a cell plate. Okay, it's a plate in the center of the cell simply. Okay, let's move on.
Teacher?
Yes.
Can you tell why this cleavage is being formed in telophase?
Okay, so like I told you, because when we're doing these, so mitosis, if you remember from my from that slide in the cell cycle, I told you that look at this slide and these are exact timelines. G1 interphase is so long, cytokinesis is so short, and mitosis is so short, cytokinesis is even shorter. So if you can appreciate over here, it is so short that it occurs in seconds. And when it's occurring in seconds, a lot of things overlap. A lot of these processes, for instance, if let's say I tell you it's so short, it's occurring so shortly, some of these are aligned, and then I say one chromosome is still randomly sitting out here, not attached. But as soon as some of these are aligned up, some of them might enter their anaphase quicker than this one, which is still going here to be aligned next to this one. Now, because these are overlapping so much, cell division, which is cytokinesis, remember how short that was? Look at this. How short is this? It's equal to one of these phases. It's not even long enough to be something on its own. So which is why it overlaps a little bit with the telophase, because the cell, see, when the nucleus is open, the cell is so vulnerable. The cell does not want to be in this vulnerable state, because what does the nucleus have? Every single protein. And if one protein is malfunctioned, for example, if you remember beta gene in in the RBC section when we were doing proteins, remember how the sickle cell disease came about? Now imagine even one amino acid being damaged during this process, then mitosis will have more harm than benefit, which is why all of these stages are at some point overlapping. So within anaphase, the cell starts pinching inwards, and in telophase, it is even pinched in more. The point at which it exactly pinches out and becomes two cell is called cytokinesis. But again, very much overlap in them. So do you understand what I mean?
Yeah.
Okay. And if there are no more questions, let's move on. No more questions?
No, teacher.
No, teacher.
Yeah.
Perfect. Let's move on and let's see what do we have next. Okay, now because I understanding microtubules can be a little bit difficult. So look at this. Look at the mother and the father. This is the pair of centrioles. Remember I showed you an exactly similar position to explain to you the 3+0 arrangement? Remember that arrangement in which there were three single noodle-like things in here and 0 inside? If you remember that, then I also told you that they were a pair and arranged at right angles to each other. So can you appreciate the right angles? And then these lines, these radiations show you that this mother centriole and the father centriole, which is going to be on the opposite end, will have microtubules extending just like this from the fiber. These centrioles will have extensions of microtubules. It will gather all the microtubules of the cell and use it just to first align the chromosomes in the center and then pull them apart in the anaphase. Okay. And even in the telophase, this pinching in is thanks to microtubules and other tubules that will lead to squeezing the cell in from the center, okay? Because the microtubules never really, the spindle fiber never really disappears. It's still working and working to complete this whole mitosis and then cytokinesis. And so this image is just to make sure you understand microtubules well. Okay, so I hope microtubules are clear how they're working, because it becomes very difficult to appreciate them in such an image. So now you have a 3D image of it. Okay, again, this is from your book. Look at the steps. Can you identify the prophase? So they've called it the early prophase, which is not exactly the prophase. It's the, like I told you, because these steps are overlapping so much, the end of interphase and the start of prophase can have an early prophase in which the centrioles or centrosomes are now moving to the ends, and then disintegration will start. As soon as disintegration starts, we call it the prophase. Movement has started as alignment and extension of the microtubules. And then look at this, they've shown separation here. Look at the separation steps. Look at how the centromere moves in first into both sides, and these ends move in last. Look at the anaphase moving in. Look at the telophase, that even though the nuclear envelope is reforming, the cytokinesis the cytokinesis has been started, not completed by started, but started. Look at the microtubules still performing their functions of pulling cytokinesis inside and breaking down. So is this step clear? This is what's happening in your book. I hope it's very easy. I hope the whole big text everywhere doesn't confuse you now when you go back and read it. So let's see, if I ask you to tell me very quickly without reading anything, can you tell me where the anaphase is? I can tell you very easily. This is the anaphase. Where can you tell me where the metaphase is? This is the metaphase, and then you can just name the other ones. Before it, we're gonna have a prophase, then we're gonna have a telophase or cytokinesis. So how did I tell you so quickly about the anaphase? Because the anaphase is the time when we see, look at these chromosomes separating, look at these spindle networks, these tiny threads pulling something apart, something that was in the center in this one, which is the metaphase, being pulled apart, which makes it the anaphase, and the rest of the other phases will become very difficult to recognize otherwise. So you can always, whenever you're given something like this, you can first look to find a phase in which everything's aligned in the center, which is the metaphase, and then being pulled apart right after that image, which is the anaphase, and then you can put in the rest of it very easily together. So will this be clear, and will this be simple and easy to label?
Yeah,
Just
miss.
need to
Yeah, ma'am.
recognize the central, and then right after it we should have an anaphase, we should have a separation. If you understand this, then can you tell me which one is going to be the anaphase and the metaphase over here? A little bit difficult, but just make sure that you're putting in your brains into it. Put your brains into it, look for something central, and then look for it separating. Okay, so which one do you think? If this is A, B, C, and D. Okay, let's A, B, C, D, E.
B is metaphase. B.
And then C is Anna.
C is
Okay.
anaphase.
Okay, so A, B, C. Okay, so C is anaphase because you see that butterfly-like network. But what you need to see next is that which one is the metaphase? Is this one the metaphase, or is this one the metaphase? Look at this one. Doesn't this one have the alignment in it?
Yes. Yes,
Yeah.
this one.
That
So
one is
I
Telophase, I think.
deliberately put in an image. This image went from here to here. I deliberately put in an image that goes from this side to this side just to see if you guys were actually listening or looking for trends on the picture. So what you guys have to look for, look at the central plate. This plate has disintegrated. What's actually happening is, look at these random fibers. These random fibers haven't yet been aligned, and the nuclear envelope still seems like it's disintegrating. It hasn't even fully because some of them are still in the center. So this is in the end of interphase and the entrance of the prophase, like I told you, happening simultaneously. So can be a little difficult on live slides because these are not sketches. Then the next one, we couldn't capture the next cell in its actual prophase, so we had to capture it in its metaphase, and then we had to capture it in its anaphase. And then this is the end of the anaphase because look at this here, it hasn't completely occurred. So this is the end of the anaphase, and these are the two daughter cells that are exactly identical. Both will have the exact DNA. And then when we're talking about DNA, so is this much clear? Can you see it occurring from here to here? Or do I still need to
Yes.
explain
Yes,
it?
Yes.
Okay, and then if you understand that these two, now since we're on this slide, if you understand that these two cells are daughter cells of this cell, then you need to understand that these two cells will have the exact number of chromosomes. How much is that in a normal cell? How much chromosomes does this cell have? How many? 46, right?
yeah. No.
And I'm telling
Yeah.
you, or let's say, so if I'm telling you this cell initially in its interphase, this cell had 46 chromatid, and then it replicated to have 46 chromosomes. The number was same. However, the DNA in this cell was just 46, but the DNA here is double. How much is the DNA here? One in this flap and one in this flap. The number of chromosomes is 46, but the number of DNA is going to be double, which is 92 something, or is it 92? I don't know. My math isn't good, and you can tell probably because I'm a doctor person. So yeah, it's something 90. Let me just multiply it. Okay, I don't want to. If this gets uploaded, I don't want people to think my math's that bad. Yeah, it's 92. Okay, not that bad, guys. So now in this cell, in the interphase, 46 chromosomes and 46 DNA. As soon as it's replicated, 46 chromosomes because they're not yet separated, and 92 DNA. But then when it forms these two daughter cells, now how many chromosomes do the daughter cells have?
46.
And you've said
23.
that because that's a fact. But do you know how? If I'm telling you if this chromosome, if a chromosome is moving in like this, if the chromosomes were exactly like this, and all of them were moved into the other place, how do you know it's still 46? How much is the DNA? Is it 92 or is it 46? So always remember that what actually happens is the cell is clever enough to make a copy of itself to form two DNAs, right? The number of DNA has changed. It has become 92, but the chromosomes are still 46, right? And what ends up happening is that because this strand goes into this cell and this strand goes into this cell, the number of chromosomes is preserved because chromosomes are essentially just one structure, whether it be a single one or a double one. It's just this one structure. The number of chromosomes has been preserved, but the number of DNA has become 46 instead of that 92. Because now it's a single DNA and not a double one. And when this cell will go back into the cycle by replicating, then it will again have 92 DNA. But that DNA will quickly again go back into the cell cycle to break down to become 46 and 46 again. So is the number count clear? Do we understand the numbers? Do we understand what's happening, what's becoming less and what's becoming more?
Yeah, so the DNA changes, but the chromosome doesn't
Yes,
change, right?
the DNA does change.
Okay.
I mean, even if it wasn't changing, then wouldn't this cell have 23? Then wouldn't 46
Yeah.
just become a lie?
Yeah, it would be a reduction division then.
Yeah. Is this clear for everyone? Any questions? I
Yes.
can explain this again. Just make sure you understand it. Clear for everyone?
Yes.
Shah Faisal, do you have it clear?
Yeah.
Any questions? Anybody has ever done a past paper that did not understand, ask it right now. We're going to do past papers, obviously, but if there's anything that's still remaining, please do ask, okay? Okay, so then let's move on. Now, how does the cell, now let's talk about this even more specialized terms. There are two structures important in this anaphase, the centromere and the kinetochore. In fact, when we looked in closely, we realized that the centromeres are two separate structures stuck together, ready to be broken apart, but they are not what the microtubules attach to. The microtubules attached to their covering, a protein covering called a kinetochore. And this kinetochore is what's pulled apart. The centromeres are preserved because we don't need them to break. What we actually break is this covering of kinetochore. As simple as that, nothing confusing. Is that clear? What do the microtubules attach to? They attach to the kinetochore. So now, because you're in A-levels, you need to know this detail. They attach to the kinetochore to pull the kinetochores apart, not the centromeres apart. The centromeres are the preserved structure of the chromosome. They cannot be pulled apart. Kinetochores are the proteins around them that can be pulled apart. Okay. Now I need to know the importance of mitosis. So we are 5 people. Let's each give an importance of mitosis. So let's start with you, Raid. What do you think is one importance of mitosis?
Repair of damaged tissue.
Repair or damaged tissue. And why do you think that's happening due to mitosis?
Due to mitosis, the damaged cells replaced by new cells, by healthy cells.
But why couldn't that happen by just a reduction division? Why can't I have 23 chromosomes in the new cell that is repairing?
Because the number of chromosomes wouldn't be conserved, so it couldn't function like a normal body cell or a somatic cell.
Very good. So we need the exact cell. We need the exact same cell to perform that exact same function. So one of them is repair. Safa, you're next. Safa, can we have one reason why mitosis is important? You can Google it. I mean, come on, you guys have your phones, you have your laptops. I'm not even monitoring you guys. So give me a reason.
Miss, like the growth cell replacement.
Okay, so Ray talked about cell replacement. Growth is an important factor. For growing up, we need, for even my hair to grow, I need more hair cells that are exactly the same, right? For my height to grow, I need more muscle cells or more bone cells and etc. So that's a good point. Shah Faisal, why do I need mitosis? Third point.
For asexual reproduction.
For asexual reproduction, I need so in humans that isn't possible. We don't do asexual reproduction, and I don't know if that's a happy fact or a sad fact, pun intended. But in fungi or in prokaryotes and other stuff, we have asexual reproduction, because sometimes because for some things that are not mammal or reptile or in our genetic materials, those organisms might find it difficult to find something exactly like what they are, because they're so small in nature and so fragile in nature. So for them, survival is highly dependent upon other forms of reproduction, which includes asexual reproduction. Okay, Aruba and Abdullah, 1 point each. I'm sorry you guys came last, because you're last in my list. So
It's okay. For me, it's the immune response. The cells they clone in, like the beta and the the beta lymphocytes. They cloning of the beta lymphocytes.
Any WBC, cloning of any white blood cell will need mitosis. Okay. Why? Because we're cloning them. The word clone means exact copy, right?
Yeah.
Okay, good one. Aruba.
Mitosis, replacement of cells and repairing of tissues. And it
Okay, so I
produces
heard that
genetically
chicken right there, which is why I'm not going to consider this point.
What? They produce
Abdullah got you in trouble.
genetically identical cells.
Sorry, can you repeat that?
They produce genetically identical daughter cells.
Okay, and why do we need them? I need importance. That's a fact, but I need an importance. Think about it. Think about it casually. Why do you think it's happening in us? Anything.
Ma'am, to make sure DNA isn't lost.
Okay, that's a very important point. We may want to make sure that the DNA is preserved, and we're going to talk about that very soon as well. But any other point, anything that comes up in your mind? Aruba, I want you to put in that mind, put it in. I'm sure you have it there.
I don't know.
Okay, that's fine. That's fine. So you can talk about replication of any cell and think in it in that direction. For instance, now let me tell you, you're not, because all of these guys have used every single term they knew, which is why no other term is left. But if I was asked, why do I need it? So I can say something like, maybe I want more stem cells. I need more stem cells replicated to make sure that I have basic progenitor cells or basic cells for any other cell that I need. I might need more cells. Why do I need identical clone more cells? I can need them because I want to preserve something. For instance, in the liver, if I want to store lots of glycogen, I might need more of those hepatocytes that has specific function of storage. Why do I need to store all of those fats that I eat? How do I store them? Do you think there's this one fat cell that's existing since you were born? No, it's mitosing and growing more of itself, just to store more of that adipose tissue, right? So you don't have to learn these. Just make sure you understand why it's happening. Why do I need a clone? So very easy, right?
Yes.
Yeah.
Okay. So let's see. Now, since we were talking about preserving DNA, let's talk about the telomeres that we also stained on immunohistochemistry in the beginning of the lecture. They were at the tips on both sides. The importance of a telomere. Now, I want you to listen to it once clearly and understand it. I told you within this chromosome, some DNA has a genetic code for a protein, and it's called a coding DNA. And some of it is non-coding. For instance, the centromere that only has a functional use of attachment. Telomeres are non-coding. They do not code for proteins. When I say coding, it means for proteins. Then what is their function? So in every replication of DNA, what happens is the cell isn't smart enough, and sometimes the enzymes get tired and forget to replicate the ends. So imagine if we did not have telomeres, every time the cell replicates into two, the DNA replicates into two. The cell only divides, right? The DNA replicates from this DNA to become this DNA. It forgets to replicate this portion. And the next time it forgets to replicate this portion, which will eventually lead to loss of important genes, which is why nature gave us something called telomeres. It's extra DNA. Right now, we don't recognize its purpose except this purpose. And this DNA, every time the cell replicates, even if it forgets to replicate this portion, we still have some excess non-coding portions remaining, and the coding portion will never be lost. Again, after many divisions, the cell enzymes again could not replicate this much. We still have telomeres remaining. Again, they could not replicate still some telomere remaining. At any point in that cell's life, when we have no telomeres remaining, that cell, these cells with no telomeres can no longer divide. And how do you think you grow old? How do you think you start losing hair? Because those hair cells, those cells of hair can no longer have telomeres. And so the cell is so smart, it knows that now if I divide, I'm going to be losing some of my DNA from here. And then maybe I become a cancerous cell, maybe I become a bad cell. So what it does is, after this point, all of these cells at the end of telomeres will declare their death. They'll be like, oh, I'm old. Now all I'm going to do is die. I'm not going to replicate anymore. If I get damaged, if I lose my hair as an 80-year-old, I'm not likely to get it back as compared to when I'm 20 years old. Even if I go bald when I'm 15, I'm going to have them back, because all of these have many telomeres remaining and many telomerase enzymes to make sure that these continue that way, as compared to when my cell has replicated so many times and lost all of its telomeres. Is this clear?